Ellen Alexander, and I'm on the board of Grassland Heritage Foundation, which is a nonprofit that focuses on prairies. And we very much are glad to have all of you here. So what I'm going to do is just give a short talk to kind of introduce people to prairies if they aren't familiar with them. Also a quick introduction to Grassland Heritage Foundation, and most importantly, an introduction to Liz, who's really our featured speaker today. So tallgrass prairies and other kinds of native grasslands really dominated the center of North America for thousands of years. And so you can see where Lawrence is on this map in the tallgrass prairie region. Probably most of you are familiar with the Flint Hills to the west of us. That's a wonderful rocky area that still has a lot of prairie. The Luss Hills is another interesting area up in Iowa. And these tallgrass prairies, then, what are they? So they're native grassland, and they do have tall grasses. So as many of you are probably familiar, you can have big bluestem and Indian grass and other grasses that can get, 6-7 feet tall, especially in wetter areas. There's many kinds of wildflowers, a few woody plants, and lots of very cool animals. So grassland birds, lots of insects, mammals, reptiles, you name it. And also microbes. And I think we tend to forget about the microbes, but that is the focus of today, is really thinking about those microbes as well. These are some of the mycorrhizal fungi that Liz will be talking about today. These are nodules of nitrogen-fixing bacteria. This is on soybeans, not a prairie plant, but they are on prairie beans and legumes as well. So what led to the dominance of the tallgrass prairie in this part of the country? It really is kind of the interaction of at least these three factors. Climate, we tend to be drier and have drought areas relative to farther east, for example. Fire can be introduced by lightning strikes, but a lot of the evidence suggests that indigenous people both set and manage fires in the area. And then that also interacts with a whole bunch of animals, especially those that are grazers. I think we're all familiar with bison. Historically, there were elk here as well. Insects should not be forgotten. Tons of grasshoppers do feed a lot on plants. And cattle in a well-managed prairie can be very important also in prairie management. But as I think people know, we don't have much prairie left anymore. So on the right here is a drawing. The light green is the tallgrass prairie that was historically present. The dark green is the current prairie. And honestly, I think this map is unfortunately overly generous because the reality is there's only 1 to 4% of the historical tallgrass prairie left today. Now, of that, about 2/3 of that is in the Flint Hills. Because there was rocky soil, the Euro-Americans, when they came, could not plow it. And so as a result, they developed a grazing culture and fire that is much more consistent with prairies. But what did lead to the loss of the prairie was a lot of activities, especially in the 1800s with Euro-Americans coming in, plowing the prairie, suppressing fires as they built settlements. They were not fond of the prairie fires that would sweep through there. More recently, native woody plants are moving into a lot of prairies. Some non-native species are a real problem. Herbicide use in prairie hay meadows is a real issue. And then just development, cities and towns and housing, etc. And we continue to lose prairies. I developed this slide for a previous talk, and it really kind of hits me because we have a progressive, environmentally supportive community. But here in Douglas County, we've lost a lot of prairies just even in recent decades. So historically, about 87% of the county was covered by prairie. Now it's less than 1%. And as you can see, we continue to lose prairies. Since Liz is going to be talking about the plant-associated microbes, I thought I'd give just a real quick introduction to some of the plants in the tallgrass prairie. My sister, Linda Forbes, wonderfully is an illustrator, so I'm leaning heavily on her work, which I think is pretty beautiful. So grasses, they really are most of the biomass, most of the sheer bulk of grass material. of plant material. And so these are kind of the big four we tend to see: big blue stem, switchgrass, Indian grass, and little blue stem, which right now is really beautiful with its uh, fruits. Wildflowers really account for the diversity of the plant species in the prairie. And if you have a well managed site, it can have over 200 different plant species. Two of the major families or groups of related plants are the sunflower family and the bean family. And those two families, plus the grass family, account for a lot of the species of the prairie. But there's a whole lot of other species as well. So all the plants here are native prairie plants, but they differ in what's called a coefficient of conservatism. This is a phrase you may hear people talk about. Conservative species are species that today are really only found associated with tallgrass prairies. So common sunflower is a native prairie plant, but we know that it is found anywhere. It can be found along roadsides, parking lots, you name it. And so it has a very low number here. On the other extreme is Meade's milkweed. This is a rare prairie plant that's kind of the poster child for prairies in this area, and that has a very high number. A few shrubs. Lead plant's a plant that many people may have heard of that we think of as a wildflower, but actually is woody. And much of the biomass or the sheer bulk of living things is underground in a prairie. This is a great picture. This is a compass plant, which can get taller than me, and you can see just the depth of its root system. And these are then drawings based on work by John Weaver, a 20th century ecologist who did a lot to show the architecture of depth of roots. Especially the wildflowers and shrubs can get really deep. So the way I personally look at it is that prairies are a natural heritage. If kind of our generation doesn't keep them going, we're going to lose them. And so the way I look at things is first priority is protect the prairie remnants, the native never plowed areas. And unfortunately, we can't just put a fence around them. We have to do appropriate management. We want to encourage prairie restorations. This is a planting of prairie plants out at Free State High School. But we also have to recognize that they don't have the full ecological complexity of a well-managed remnant prairie. It's also great to have prairie plants along our yards. That's something all of us can do. They're beautiful, they're tough, and they really provide food for insects and plants. And finally, spreading the word. And so my interests align very well with Grassland Heritage Foundation, the group that's sponsoring this. Our group is focused on land acquisition. We have prairies of our own that we manage, stewardship and restoration, and again, public outreach. And these are some of the things we do. Perhaps people know us best for our native plant sales. We also have public talks, a variety of stewardship and restoration. We also give away money, graduate student research funding, and we also have a fund that helps groups that have already set up a prairie maintain that prairie at a school, church, or whatever group. And we always love to have new people join us. So if you haven't spent time in a prairie, do so. And it might be fun to do it with people that know prairies, because prairies are subtle. They're beautiful areas, but a lot of times when people first walk into them, not really sure what they're seeing. So without further ado, let's have Liz. But I first want to yell out to Kathy, who's somewhere back here, way in the back. Kathy has done a lot to help set this up, so I want to acknowledge her help, and she has various things to look at over there. But Dr. Liz Posel is going to be talking next. Liz grew up in Indiana, and she did both her undergraduate and graduate work at Indiana University. She always had loved fungi, did mushroom hunting with her dad, but when she was involved at Indiana University, she got studying these mycorrhizal fungi in the soil. That was her start into this. About 10 years ago, she moved here to University of Kansas, and she has done research with these mycorrhizal fungi in lots of different ways. She's worked with the Land Institute out at Salina with perennial agriculture. She's been involved with local farmers and specialty crops. Most recently, she has worked out at Johnson County with some of their large-scale prairie restorations. So I have a wonderful list of the ways that she thinks of herself that I thought I'd share. Officially, she's an assistant research professor at KU, but other names are mycologist, person who studies fungi, microbiologist, makes sense, ecologist, fungal library curator, that may not make sense, but ask her about it if you're curious, plant community ecologist, agronomist, and entrepreneur, because she started her own company and she has some things about that here. It's a wonderful way to use this inoculum on garden plantings that you might have. And proud childless cat lady. And she also has shared that she loves prairies so much she willingly spends her summers in them, even in the Kansas heat. And finally, I think I needed to say that her favorite snack food is chili cheese Fritos. So without further ado, Liz.
How are we looking? Great. Well, thanks everyone for coming out today. I just wanted to share a little video I took of everybody at the Grassland Heritage Foundation Native Plant Sale in the spring, where we had a Michael Bloom vendor booth busted the door to pick up their native plants. So this is a really cool community. So thanks for caring about native plants and for coming to hear about some native soil microbes. So soil is a massively complex ecosystem. I really like this figure because it shows you how crazy it is down there. All these arrows of these different organisms interacting with each other, facilitating, competing, eating each other. It's really complicated down there. But today, mostly I'm just going to talk about this chunk of what's happening in the soil. Plants, their roots, and fungi. We'll keep it simple. And so one of the things I call myself is a mycologist, which people often think that I'm doing this, which I do. Sometimes I like hunting mushrooms and playing with them. I don't really enjoy eating them that much, but they're fun to look at. But for my work, the type of fungi I study are called arbuscular mycorrhizal fungi, and they're different from these in that they're microscopic. So you pretty much need a microscope to see them. Although there are some you can see with the naked eye, and I have some examples of that today at the little science table we can look at later. And so arbuscular mycorrhizal fungi is a mouthful, and usually I just call them AMF for short. So if I say AMF, that's what I'm meaning. And these fungi are ancient. It's thought that they developed about 400 million years ago. That's when the earth kind of looked like this. So there's little teeny tiny fern-like plants all over. Animals had just figured out eggs and were leaving the ocean, and so were the plants too. So fungi are right along there with them as plants left the ocean. And we know that by looking in fossils, where we can see some of the structures of these fungi in these ancient fossils. And today, this is what these fungi look like. They have sort of two main forms. Up top is a picture of their spores, and the different sizes and colors and shapes is how we tell the species apart. There's about 350 species described so far. And a lot of the function of these fungi lies in their hyphae, which is sort of depicted by this white stuff spreading all out over the soil. It's this filamentous and interconnecting network that spreads all throughout the soil. And that's where a lot of the function of these fungi come into play. And so plants and these AMF interact with each other in a symbiotic way, meaning both partners are getting something out of it. And so this mutualism or this symbiotic interaction begins when a plant seed germinates in the soil and the little plant rootlets interact with that hyphal network. And so the fungi actually grow inside the plant roots. And essentially, the plants are feeding these fungi carbon. The fungi can't get carbon on their own. They have to be fed by associating with this plant host. And then in return, the fungi use that hyphal network to collect nutrients, especially phosphorus, zinc, some other micronutrients, and they can provide that to their plant host. And so in associating with these fungi, plants can actually grow larger to that increased foraging area. And so this interaction is really widespread. About 85% of terrestrial plants associate with this type of fungi, AMF, including almost everything we eat, except for brassicas. Our broccoli and cauliflower, mustards don't associate with these. And almost all prairie plants associate with AMF, so they're really important for this ecosystem in particular. A little bit more about the biology of these fungi. Another complicated word, endomycorrhizal. That's the type they are. Endo meaning within, myco means fungus, and rhizal root. So these fungi are actually living inside of plant roots. And if we go out into the prairie and collect some plant roots and look inside of any root, it's going to be absolutely loaded with fungi. And so here is an example of this. This whole thing here is a plant root, and we put a special stain on this root that turns fungal structures really dark blue. So all this dark blue stuff in here is fungus, and you can see it's just loaded with this hyphae running along in between the plant root cells. And then these blotchy structures are called arbuscles. And that's where the fungi get their name from, arbuscles. And it's a site of nutrient exchange. Essentially, it's just branching out in all directions, trying to increase surface area to exchange nutrients. These are other examples of branchy things in nature. Arbor, like arbuscule, a tree, does the same sort of thing to capture sun. And then our lungs alveoli, branch out to collect oxygen. So similar strategy, but for fungi inside roots. And these fungi do a lot of things besides provide plant nutrients, although they're mostly known for that. For example, all of that hyphae in the soil is really good at binding soil together and can help with soil erosion issues, for example. The fungi themselves are carbon, and some of the fungal structures are really tough and can stay in the soil for a long time. So it's a type of carbon sink too. I have some examples of spores at the sciencey table. We can actually crush them and hear them break. So they're really tough structures and they can stay in the soil a long time. Also, fungi can prime the defense system of plants, which can help them from pests and herbivores and that sort of thing. So they do actually quite a bit in the soil. Thinking about how these fungi move, they have a couple of different strategies. One is just growing through their hyphae very slowly in the soil. So for example, there's some fungi associated with this plant here. Its little hyphal network can grow along and interact with the next plant. This process is really slow, and when we track how quickly these fungi move, it kind of depends on the species. Some don't really move. If we inoculate them here and we track how far they're spreading, some don't spread very far at all. And some might spread 5 feet per year, for example. Other ways they move around is in the wind and the water, but often if they're in the wind, they're kind of dead because they don't like the sun. They're subterranean organisms, so they can be solarized to death. But sometimes a spore might survive in the dust, for example. Another way they move is by little critters, even us in the prairie, probably my shoes today. Deer, bull, those sorts of animals that are digging around and getting dirty can move spores of these fungi around. That's sort of externally. And there's been some fun work looking at the internal spreading of AM fungi in the guts of worms, where they might eat some spores as they're grazing in the soil. And these spores are full of fats, they're like delicious little french fries. And some get broken in the gizzard and digested, others pass through the whole worm and then come up in its poop, and those spores might germinate. So that's one way they can move around, but still pretty slowly. Another way we move these AMF around is intentionally through inoculants. So I'll be talking a lot about this today, both thinking about some products and then some different ways our lab has tried to move these fungi around. So I tend to study mycorrhizal communities and these two types of soils. In the tallgrass prairie, particularly untilled prairie, old growth prairie, versus in more disturbed environments. This is a picture of an ag field that's sort of in its dormant stage. The corn was removed and it's a brown field at this time. What we see if we look at the mycorrhizal communities below these two soils in the prairie, there's a ton of AMF there. There's a lot of species, and the ones that are there tend to be really beneficial. There's specialist species as well. In disturbed soils, we tend to see fewer AMF, and the ones that are there are less beneficial, generally speaking. And because they're so dispersal limited, they can be really, really slow to recover. And the reason for that is the way we treat our soil. So for example, things like tillage can chop up those hyphal networks and result in the death of these fungi. In things like our yards, if we're planting a seed-free grass or a plant that's not a good host for these fungi, that can also lead to lower abundance of these fungi over time. So Helen showed a pretty similar picture to this, but I like this one as well. It's looking at the root architecture of different grassland plant species. And you can probably recognize some of them, like a big blue stem with its turkey foot flowering head, has a long root system with a ton of fine roots, versus this echinacea coneflower has, maybe a slightly shorter system and not very many fine roots. And so it's thought that the root architecture of plant species really can determine how reliant it might be on a fungus. And it's thought that these plants with coarser root structures, fewer fine roots, really rely on the fungi and that hyphal network to do the foraging for them. But a lot of the work I do in the greenhouse is looking at how different plants respond to the fungi. So essentially what we do, we take fungi from the prairie and grow plants with, as indicated by the plus, and without that fungi, and look at the growth response in just a weight ratio. So if dahlia up here is growing 10 times larger with fungi than without, it would have a mycorrhizal response of 10. If panicum here is growing about the same with and without fungi, it would have a mycorrhizal response of about 1. And so if we look at mycorrhizal response across a successional gradient, based on coefficient of conservatism score, which Helen mentioned, where on the left are very early successional plants are non-native plants and weedy plants like Helianthus annuus, the common sunflower. And our plants over here tend to be only found in undisturbed prairies. We kind of see this pattern here. Every little dot on this graph is a plant species, and the general pattern is the weeds, the non-native plants, they don't really care about their prairie AMF. And the late successional plants are highly dependent on their native mycorrhizae. So I think about this figure a lot, and we use this to predict what might happen in a restoration. So let's say we took seeds from all these 25 little dots, all these plants, we collect them, or little seedlings, and we put those into a restoration environment that we know has disturbed fungal community. What we would expect to happen if we throw those seeds out there, the plants that don't care about fungi would establish really well, and the ones that are really dependent on their native prairie AMF would be limited and not be able to establish. And that's kind of what we see when we think about restoration and the challenges we see in restoration. A lot of these late successional plants really have trouble reestablishing in our prairies. And so the goal for a lot of my work is to try to figure out how to get these guys back into our restorations. And so this mycorrhizal response is something we work out a lot in the lab and at the Kansas Biological Survey. And we have a database of about 200 different plant species and the response to their prairie fungi. Back at the microbloom table, it's over there. You can take some pictures of it if that interests you later. And so we began to take these sort of ideas about plants being really dependent on their native AMF and implementing them in restorations. And what we do is something called a microbial transplant. So we're taking microbes from an old prairie, an undisturbed prairie, and putting that into a restoration. And that microbial transplant, we often call that inocula, so another sciencey word. There's a couple types of inocula I'll be talking about today when I get to my case studies. Mostly I'll be talking about native inocula. Whole soil is as simple as it sounds. We go to prairie, collect all the soil, move it around. It's really easy to do. It's hard to find soil to move around though, so that can be the limitation of that strategy. I also work a lot with guild-specific inoculants. For example, just isolating the AMF from that soil and culturing that up, and I'll talk a little bit about that later. There's also a ton of non-native inoculants available. You can buy these things anywhere, Amazon, Home Depot, Ace. There's guild-specific inoculants for mycorrhizae, rhizobia, and sort of these kitchen-y sink products that some claim to have more than 800 different species in them. And I'll talk a little bit about those today too. But first I want to oh yeah.
You want the whole question to the end?
I think I'll have like 3 chunks and maybe we'll have 1 question for each chunk and then at the end we'll have a bunch. So I'm almost at the end of my first chunk, so I can tell you're ready. But I want to describe a little bit about how I grow these fungi and how I've acquired the ones I use for my experiments. And finding them is the fun part. And so you put on your favorite field clothes and you go out into the prairie and take a soil pour. And so in Kansas, there's tons of prairies to choose from. You can just go over to Kansas, there's acres and acres and acres. For some of the places I've done my research, prairies are much more limited. And for example, the only prairies in southern Illinois where I did some of my research were these sort of cemetery prairies or railroad prairies that happen to be not disturbed, but they're very tiny. So after we take our soil core, we pound this thing into the earth. We have some dirts. We then run that dirt through sieves, which is kind of like a colander you would use to strain your macaroni and cheese. And basically, it's just a thing with different size holes. These holes are so big you can see them. These ones are so tiny you can't. But we sort of look at the size of soil we know to be mycorrhizal spores. Sorry, that should be micrometer. They're about 40 to 400 micrometers, so they're real tiny, but we can see some with the naked eye. And we'll get to look at some of these spores later today. So after we have that size of the soil that is spores, we spend loads and loads of hours at the microscope sorting them by species based on their color and shape and size. Big yellow, tiny white, that sort of thing. Growing them is pretty challenging, so we have to grow them in soil, but soil has a bunch of stuff in it, right? So first we have to sterilize the soil, usually through autoclaving. After the soil is sterile, we can put our one species of fungus in the dirt, and then because these plants are obligately dependent on, the fungi are obligately dependent on plants, we have to use a plant to culture the fungi. So we put some little prairie seedlings in the pot. We allow it to grow for 6 to 9 months. And then at the end of the day, I'm taking all these pots with different isolates of fungi, mixing them all together, and this is like the inoculant that I'm using in my restorations. So this whole process takes a really long time. To acquire the fungi, it usually takes about 2 to 3 years. Think about it in terms of starting like a native seed nursery, right? It's a lot of work, and we need a lot of specialty equipment to do this, so it's really time consuming. Even once we have fungi in collection, it takes about a year to produce enough to do a restoration experiment. So that's how we grow them. I also wanted to just throw out some ways that people think you can grow them that you can't really grow them. One being compost teas and other being compost. And so those methods are great for growing some microbes, including fungi, but just not this type of fungi because these fungi need a plant to give them sugar. They can't find it any other way. So that doesn't work really well for growing these fungi. And then in terms of applying these fungi, sort of putting them on the surface doesn't really work either. And I'll talk a little bit more about that later. Okay, so I've finished the biology of AMF section. After that, I'm going to do some sort of case studies in the prairie about how we put these out in restoration and the effects we see. And then I'm going to talk a little bit about microbloom and connections to industry and INRAM. So I'll take one question at this first chunk if it's relevant to biology, if anybody has one, or we can wait until the end.
One question about the worms.
Yeah.
You said earthworms, you meant like the native earthworms, not like giant red blue.
I think probably most worms can move them around, maybe not the nematodes, like that kind of thing that is almost a worm, too tiny, but yeah. Yeah, it's hard for them to move, which is why we spend a lot of time figuring out ways to move them. So I'll jump into the next section, ways we move fungi around. The first thing I wanted to talk about was the differences between native mycorrhizae, which we culture from prairies and put out in prairies, versus commercial products. And ChatGPT made this funny image for me about commercial products, but they do kind of actually look like that. The difference being commercial products usually aren't native. Sometimes they come from other continents. Sometimes they don't tell you where their fungi come from. But it's a huge industry. There's tons of products you can buy, and there's $1 billion globally spent on these commercial inoculants. So how do they work in prairie restoration? I've done quite a bit of work on this, but I just wanted to show a couple little case studies. The first being an inoculant that I bought at Prairie Moon. So probably most people in the room, if you're interested in native plants, have heard of Prairie Moon. They're like the biggest seed producer in the Midwest for prairies. And you can go and Google inoculant on the website and find something, and they'll sell it to you. It's not something that Prairie Moon makes. They just sort of post the sale for this product. But it's $19 a pound, put it in your cart, and it will come to your house at the end of the week. So I wanted to see how this product compared to our native mycorrhizae products. The closest application suggestion I could find on that package was about 5 pounds per cubic yard, which when I do my little mycorrhizal response, it's in a tiny cup, like a tiny pot, right? And so the equivalent would be about a 25th of a teaspoon per pot was your application suggestion. Which, if anybody bakes or has ever seen a teaspoon, you know that's really small. And a 25th of that is like, I don't even know how you'd measure something that tiny. So for our mycorrhizal response assays, we're usually using like a tablespoon, so 75 times this volume. So what I did for this experiment was sort of took a middle ground where I did a whole teaspoon for both types of inoculants, that Prairie Moon product and our native inoculants. And this is kind of what it looks like when we're inoculating plants, either for mycorrhizal response assays or if you're putting out in the field. What we have is our usually long skinny pot. We fill it with some sort of sterile soil. We put about 10% by volume of that inoculant, usually a couple inches below the soil surface, because those microbes like to be below ground, so we want to bury them in a little bit. And then we put a little seedling, we plug, we plant it in, and let it grow for usually, depending on the experiment, 10 to 16 weeks, and then look at the results. And so here's some pictures of that. On the left, Baptisia, our legume. In the middle, Echinacea, our composite. And on the right, big blue stem. Then in each little figure, native is our native prairie AMF from Kansas, the Prairie Moon product, and then a non-inoculated control. And it's pretty easy to see just with your eyeball that, for example, Echinacea didn't really like the control or Prairie Moon product, but did okay with the native. Same with Baptisia. The big blue stem sort of had an intermediate response to this inoculant. So that's really easy to see with the eyeball, but here is the same pattern in bar graph form if you like bar graphs. I do. But essentially, this intermediate response was interesting to me. I thought perhaps it's a response to fertilizer. If you look at the little picture, these little big blue stems are pretty dark green, and a lot of these commercial products have labeled and sometimes unlabeled fertilizers. And so I didn't test this in this experiment, but based on how happy and dark green this plant is, perhaps that's one reason. Another reason is potentially there's only one fungus in this commercial product, and maybe this big blue stem just happened to like that species of fungus, and Echinacea didn't like it, and Baptisia was actually inhibited a little bit by that species. So that's different than our native inoculant, which is usually quite a few different species. But this pattern of plants actually doing worse with an inoculant has been seen before. Here's some data I did for monarchwatch when I first moved to town with Peggy Schultz. And this is looking at a different commercial product, but one that RES uses when they grow their seedlings. And on average, these 4 milkweeds didn't like the commercial product, and they liked our native prairie AMF. And people from labs all over the place have found this same sort of pattern with their local inoculant. Different experiments finding that native microbes work best for restoration and commercial products don't. So the question becomes why? Why do these products fail? Like plants, these AMF can be locally adapted. So perhaps it's a site and regional mismatch, like these products aren't native. That's one idea, and I would love to test it. But unfortunately, most of these products are dead. So there's a global meta-analysis looking at 250 inoculant trials, and they found that 87% of them, these products were unviable, so they don't colonize plant roots. So we can't actually ask very many questions with them because they're not viable. Zooming in on the US, this is some data from products you can buy here at Amazon. We found the same exact pattern. About 87% of them were non-viable. Some didn't have any fungi in them, dead or alive, which was very interesting. And the ones that did have fungi, it tended to be way lower than they said. You know, they might say 3,000 spores per gram, and they had something like 15. So off by orders of magnitude. So long story short, the AMF inoculant industry is a sad place, and I could talk about that for hours, but I won't. But the lady from Up From Dust, Celia, interviewed some of the lab members about these papers that we worked on, and it's a really fun listen. She did a great job, and you might have heard snippets of it at NPR or KCUR. Yeah, you should totally check that out if you're interested in learning more about commercial inoculants. But that's all I'll say for them until I talk about my own business a little bit later. In the meantime, I want to talk a little bit about native applications of fungi in restoration, focusing on some of the long-term experiments that we've done and how we're thinking about moving this along is a science from, the plot scale to the acre scale. How are we going to do this? The thing that I'm really interested in for my restorations is biodiversity. So we can see with these 25 plant species, how do we get all 25 of them to establish? And I think the key to that is moving these microbes. So that's what I'm trying to see, if we can move the microbes to get all these plants to establish. The first case study I wanted to mention was one we started in about 2012, looking about what kinds of inoculants are the most important to include in a prairie restoration. So for this experiment, we had little plots where we had no microbes, so just plants. Of course, there was whatever disturbed microbes would have been at that site previously, are still there. And then we added to those whole prairie soil, which would have anything in a whole soil from AMF to rhizobia, worms, all the good stuff in prairie soil. And then from the same whole soil, isolating just the native prairie AMF and excluding everything else. And so we installed our inoculation treatments on the roots of little seedlings that I kind of described earlier. And for this study, we followed about 3,000 little seedlings and restorations in Illinois, 2 in Kansas, and 2 in Oklahoma. We also seeded all the sites with a diverse seed mixture of 40 to 60 species. And so I'm not going to talk about each restoration individually, but kind of the general patterns that we found. And first, I want to talk about what happened to those little seedlings, those little native prairie plants we inoculated. And here in black is our native prairie AMF. In dark gray is the prairie whole soil, and in white the control. And up top is survival, and down below is productivity or leaf number. So you can kind of see, looking at these bars, that both types of microbes were pretty similar. They both improved plant survival and improved plant growth. And this was year 1 data across all these sites. We followed these seedlings for 3 years, and this pattern persisted over time. Looking at what happened with the seeds that were recruited next to those plant species, we saw that diversity, which is kind of thinking about the number of plants, the richness, and their evenness, diversity was improved with our native AMF, and the weeds that were present was reduced with our native AMF relative to the 2 kinds of other inoculants. So native AMF seemed to be a little bit better for the whole community. We only followed one of these sites a little longer than this. We followed 1 through year 8, and these patterns persisted. We're still seeing microbes improving richness and reducing weed establishment in these sites. And so these sites were inoculated 1 time 8 years ago, and these patterns of community response are still persistent 8 years later. And that's great for some places like Illinois. I mentioned, we have to go to sample our native AMF in these teeny tiny cemetery prairies, because in Illinois it was a lot of prairie, now it's less than 0.01%. So for places like this, we really can't think about using whole soil as a restoration tool because there's just not enough available. This study showed we can culture just the AMF from this whole soil, bulk that up in the lab, put it back out, and it can be a restoration tool. So yeah, that was really exciting. So in most of the words I gave Helen to describe me, none of them are really about plants. I don't really care about many of these other things other than they're cute in the prairie, such as insects, bugs, birds, all of that. But we know it's an important part of the ecosystem. So I wanted to think about some of these other things as a success for restoration. And the first stab at doing this in one of these experiments was looking at the Coleoptera, the beetles. And one of the reasons for that was we had a postdoc in the lab, Eric, who was really interested in beetles, so that was helpful. Logistically, they're really easy to catch. We just dug in some cups in the ground, and the little beetles walked into them, and we caught them. It was pretty easy. And then they're really functionally diverse. So we caught 27 species the first year, and here's just pictures of 5 or 6 of them, and the different trophic roles they play, from predators, detritivores. They do a lot of things in the ecosystem, so they're probably pretty important to assess its health. And so we're still working on this bug data. It's pretty fun, but I wanted to share one finding, which was that plant diversity, which is, as I showed in the other slides, responds to native inocula. The more inocula you have, the higher your diversity. Also, as diversity increases, your beetles increase. So it's kind of this cascading effect where you can inoculate year 1, seed plant community is improving over time, and that can in turn affect the other ecosystem members, such as beetles, spiders, etc. So yeah, this has been really fun to work on this project over time, and it's a rare treat to have a restoration you can visit for 8 years. It's pretty rare. So the next case study I want to show is another long-term study that I started in Lawrence right when I moved to town in 2017. This line of inquiry was thinking about if we know native AMF are probably the most important guild of soil microbes to include in the restoration, how do we do it? How much do we need? So what I did, it was an old brome field, about 20 years brome. We tilled it up, we put a diverse seed mixture out, and then I did little 10 by 10 foot plots where I put different amounts of this mycorrhizae inoculant in, ranging from none to what I like to call handfuls per acre, to the equivalent of a dump truck load per acre, just loads and loads of this inocula, to try to see where we start to see a response and maybe where that response stops improving over time. And so this has been another long-term study. Both of these, the first few years have been published, but I just wanted to show you some old pictures. The first year, most prairie restorations looked pretty weedy, and that was very true. And the first year, we needed quite a bit of inoculant to see an effect. It was about 500 kilograms per acre, per hectare, which is pretty equivalent to pounds per acre. By the fourth year, starting to look more like a prairie, and that number declined, where we're needing less inoculant to see an effect. Here's what they look like pretty recently. They're really cute little prairies. They're really diverse. You can just look at them with your eyeball and see, the different leaves and flower shapes just at one point in time here, and see how diverse these prairies are. And that diversity is related to the inoculation treatment. And so I'm going to show you a couple graphs. Hopefully it won't be too scary. My response is going to be how much inoculant put per plot, none, all the way to dump truck loads per acre. And then on the Y, I'm going to be showing you first floristic quality index. And this is a diversity index that a lot of land managers use. It takes into account richness and coefficient of conservation scores. You don't need to really know all that. What you need to know, the higher the FQI, the higher quality of the plot. And so there's this strong relationship with native inocula. The more you put out there, the higher quality the plot was. And there was about a 20% improvement with the highest densities of AMF application. Jumping into what plants are responding, here in blue is our late successional plants. Remember, those are the ones with the higher mycorrhizal response. In the field, as we add more and more AMF, we see more and more of these species. Their richness improves about 40% with those highest densities of AMF added. In orange, we see the opposite pattern. So those are our non-native plants. And as we add more native AMF, we see non-native plants decline. And I'd like to see right about here, 128, non-native abundance is kind of low and sort of stays consistently low. So we started to use this in our future trials as the recommended application rate, about 128 kilograms per hectare. So we saw both late successional plants doing great and our weeds not doing as well. What plants we're establishing? All the cute ones. We saw lots of legumes and wildflowers, and some spring ephemerals were more likely to establish with inocula. Things like Baptisia, Tephrosia, lead plant, and Spiralis is a grass that really likes native AMF. Most grasses are kind of facultative in their response, meaning they can go either way, but Spiralis tends to like it. So yeah, lots of plants that are really important to pollinators seem to be the ones responding. So to summarize what we see with these long-term studies, which are really rare, diverse native AMF applications improve seeds and plants when we put it on them, when we're planting them in prairie. The plants that tend to benefit the most are our legumes, our composites, our pollinated plants. And we do see cascading effects in the ecosystem on things such as bugs, for example. And then going forward, this key result is that we are thinking about 128 kilograms per hectare, which is, 115 pounds per acre or something like that, is the number we're thinking is what it needs to be applied to see both non-native reduction and late successional improvement. Yeah.
How are you applying it to get it below the soil?
Yeah, so we tried a number of different ways. The next case study is all about that. But in that study I just showed, we tilled it in. So we surface applied it and tilted it. Sorry if I didn't mention that. And then the first study was with little inoculated seedlings. So we tried a ton of different ways so far. Yeah, so I'll summarize that a little bit too. Thinking about how we put it out there, that's the next case study, wants to jump into that. How do we scale up from these little plots to think about acres and restoring acres with AMF? Because not everyone wants to till their site. You know, tilling's not great for your soil, can lead to erosion issues, release weeds, so that's a complication. Not everyone wants to plant a bunch of inoculated seedlings. Although I'm working on a project now where we're putting out 400,000 inoculated plants. So it kind of depends on you know your partners and who you're working with, what's possible to do at your site. So we wanted to ask what other ways can we put this inoculant out there? And so this is a set of experiments that I've participated on with Johnson County Parks and Recreation District in Southwest Kansas City, thinking about how we can use tools that are available, you know, at ACE or that land managers have to put this inoculant out there. And in these studies, we're kind of testing an inoculation rate we think that is successful based on our past studies, but using these new tools. And so here's the crew that's really helped me with a lot of this work, although it's not inclusive of everyone. But so what we did, my role, I grew the AMF and had some inoculation rates and ideas how to get it out there. Their role, they prepped all the sites, bought all the seeds, they chose the equipment to use to install the inocula, and then defined success and failure based on what they were hoping to put out there. So this is really fun. They tried testing a number of different ways to surface broadcast the inocula. The least effective being spreading it out. It's actually pretty heavy stuff, so it doesn't spread too well by hand. Some toe behind implements that simply applied too much, so they mixed those right away. But a couple that they found to be successful, there was a stand-on spreader sprayer thing called a Z-Flow or something that they really liked. And then this thing at the bottom is like a salt thing, you know, when your sidewalks get salty. It spreads the inocula pretty well. So they like those for surface broadcasting. For thinking about ways to move that inocula below the soil surface, they had a couple different ideas. Up top is a harrow. So it's sort of like this thing with these tines you can put behind a tractor or Kubota or whatever. It sort of like rakes the soil a little bit. And so if you put your seeds in inocula down first and then drive this thing over to rake into the top layers of the soil. The other method they wanted to try was a seed drill. And so lots of large-scale restorations put seeds in this implement that you tow behind a tractor called a drill. And essentially a drill, what it does, it has a little furrow, so it digs a tiny row into the soil. Then it drops stuff down at a programmed rate, inoculate and seed into that furrow. Then at the back, there's this wheel that closes the furrow. So it's a really great way to put stuff slightly below the soil surface, which we thought might be important for these mycorrhizae because that's where they live. And so here's the crew driving real fast, installing all these different methods, drilling, spreading, spraying, crushing, just trying everything we can to put this stuff out there. And so these experiments are only in their first year, so we don't have a ton of data, and you're probably tired of bar graphs at this point. So I just kind of wanted to summarize what we've seen with this first year based on the data. But this one's at Bull Creek, yeah. We also had one at Shawnee Mission Park, so it kind of spread out a little bit. But yeah, good eye. But in general, what we're finding is that there's many plants that are establishing with AMF alone tend to be wildflowers. So we saw 19 species that only established with inocula, and 16 of those 19 tend to be these wildflowers, both early and late successional, but plants that we really want to see in our prairies. So this is the same kind of thing we're seeing over and over again in these restoration trials of pollinated plants like AMF. And thinking about the response of these different kinds of treatments along the continuum, in Big Bull Creek Year 1, tons and tons and tons of weeds in many of the non-inoculated plots. You can see one little Rudbeckia, I guess it counts. But generally, the control plots were dominated by weeds, and very few seeds came up this first year. Remember, this is just year 1 data, so this could change over time. The broadcast inoculation treatment was actually pretty similar to the control. We didn't see any different responses for the native plants relative to the control. So this is throwing the inocula just on the soil surface. Broadcast and Harrow, remember that was that thing that kind of raked it in a little bit, seemed to work a little better. It was patchy though. Some plots there was a lot of native plants, and other plots it seems like it didn't work quite as well. So it was kind of an intermediate response, although it was significantly more native plant establishment with Harrow. And they put like, I don't know, 10 pounds per acre of Echinacea pallidar, some like ridiculously high number. They were really excited about it. And that one did respond too, so they were happy about that. By far the most effective thing we tried with Johnson County was that drilling inocula. So remember that was that tool that buried it below the soil surface. We saw the most native plants established and the fewer weeds in these plots. And so thinking about all these different ways I've talked about today, the ones that have big effect tend to be things like seed drills, tilling it, putting it in plugs, finding some way to install that inocula below the soil surface seems to be the key thing when we're thinking about how to use an inoculant. And
did they include the inocula at the same time as they were planting, like if you need a seed drill, mix them together?
Yeah, I think they did the seed, they drilled all the seed first and then drilled the inocula behind it, just in the inoculate plots, yeah. But I've worked with some experiments where they mix them all together, so it kind of depends, but they just wanted to keep it separate. Any other questions on this trunk or the previous part?
I have another one. Have you tried liquid aqueous application?
Great question. And you can buy a ton of products that are liquid based. I have some spores over there on the science table you should check out. Spores often, when they're in water, will germinate and grow. So that is kind of a complicated thing about their biology. Although you can buy products that are liquid that have microrhizae in them, probably they have already germinated and they're dead. That's kind of what we see in every liquid product we've ever tested. We haven't found one with viable AMF in it. Not to say it doesn't exist, I just haven't ever seen it yet. Yeah.
Have you guys done any experiments as far as like the depth of the AMF application and how that has an effect on its effectiveness?
That's a fun idea, but no.
No?
Yeah, usually we just set the seed drill, as deep as it'll go, and you till as deep as it will go. But yeah, I haven't experimented with that myself, but that could be fun. Yeah.
Back to his question about mixing with the seed or not, or would you expect any difference if it's not seed direct contact prior to application?
Yeah, I think mixing it with seed would be great. The trouble is some seeds are so fluffy and some seeds are so heavy, So like a lot of times in these seed drills, there's several boxes with like different heaviness of seeds. I think it could work well to be mixed in with like, those heavy legume seeds. Then you have your light fluffy grass seeds in a different box. And that would work really well. But yeah, in terms of installing that inocula, I think that's probably the best way to do it is to drill it right next to your seed.
So does the timing, since the seeds haven't germinated, there's no roots for the fungi to like coexist with, they can persist for however long it takes for them to germinate?
That's another great question. So for the Johnson County drill project, we planted it in April. And so the seeds were, going to grow immediately right after that, and also the weeds, which can sometimes be the host for the fungi. So that experiment, it wasn't a worry. But yeah, a lot of times people will drill seed in November, and they want to know, can they drill inocula in November, or should they wait until the spring? I usually tell them to wait if they can, because we haven't tested how well it works drilling in the fall versus spring. These fungi can be dormant, they overwinter in our native soils, so potentially. And I think they could work drilling in the fall. That's just something we haven't tested as much. I only have enough energy to do about 5 side projects a year. But yeah, something I would love, and it's definitely on my list coming up.
So knowing that the experiments with distributing into the soil are only in their first year, have you seen any indications that the bigger hurdle might be production of these specialized mixes or actually distributing them into the soil? Do you sort of have an idea?
I think they're both really challenging, right? So for my side business, me and Steve, back there, we spend a lot of time producing this stuff, and it's really hard and really heavy and hard efforts. So So that is a challenge. There's lots of things we don't know about like inoculums generally. How local is local? So usually I try to sell microbloom and I recommend it to people originally, but you know, they'll like buy it and I put it out in Washington. How well does it work there? I don't know. So that's another question. There's a lot of the science that still hasn't been worked out. You know, you can buy a product and it will say, put it in the liquid solution and put it on the soil surface. And it doesn't seem like that would work with the biology of fungi, and I'm not sure why these products say that. And they don't often have data like to support that it works in this way. They don't ever provide data. So,
Advertising.
yeah, it is mostly advertising or just like guesswork or someone said it once and now everybody just keeps saying it. But like it might not be true. So yeah, there's just a ton of science we need to work out in terms of like optimizing it as a tool. But we've gotten pretty far. Putting it underground is good. Granular inoculants seem to be good. So that's where we're at.
So are there any guidelines or government regulations of the product being what it says it is?
So in the U.S., there's not really, yeah. You can move microarrays in and out of the U.S. from any country, and it's not regulated in that way. In some states, for these AMF, there's label requirements, like you have to say what species and what the concentration is. But there's zero enforcement of that. It's like a company self-report that. And so some companies report things like we have 3,000 spores per gram of these 3 species. And then we test them in the lab and it's like you don't have any of spores per gram. And there's no one to like call them out on it. There's no enforcement of the regulation, which is why 87% of these products are not viable, right? Because there's nobody to be like, this is bad.
But there are regulations in place, there's just no enforcement.
Correct.
And the regulations were put in place by,
Like in the U.S., it's state by state, so not every state has it, and yeah, it's state by state.
and what organization it would be?
Good question. I don't remember the name. No, it's not USDA, but I don't remember the names of the different groups. And not all states have them. Like in Kansas, I sell my inoculums without labels. Do that. Here.
I'm wondering about some of the property we see where the soil has not been disturbed, but maybe through grazing, overseeding, fertilization, the soil, the native prairie soil hasn't been disturbed, but it's now almost like broom field, right, grasses. Have you looked at the mycorrhizal community of what's existing there? And then how might adding from an old growth prairie affect that kind of land?
Yeah, I think it depends on the exact things that are added. But we've done some experiments looking at like adding nitrogen over time and fertilizer over time. We do see reductions in colonization. So the plants are like experiencing a cost from associating with AMF over time. And some plants might not associate with them over time if you're able to take fertilizer out. And so all that would lead to a loss of fungi, a lower abundance of fungi over time. And yeah, I think those places potentially could be good for amending with microbes. I think it might also depend the legacy of the different like nutrient additions that were there. We have like a ton of inorganic phosphorus still. Maybe AMF would be as effective if it's something that sticks around a lot.
Along those lines, the Nature Center, Park Nature Center got sprayed. It's an intact native prairie. Any ideas on how it seems like it's bouncing back very well.
That's what I've heard. You ever visited a Here Grows lab?
Yeah.
Okay.
And I didn't know if you had any samples, whether that you could see any changes over time in terms of whether there was a dip in the AMF.
We haven't looked at
with
AMF
resilience.
there. Well, we do know that glyphosate can lead to lower spore abundance in the soil, like directly. So potentially, but maybe with one spray that didn't work so well because things made it. I don't think it was a huge loss.
It doesn't. I mean, it's really bounced back. It's very resilient. Pretty amazing.
Okay, I'm gonna go to my last section, then we can get back to some questions. But that was a fun break. The last section is real short too. All I wanted to say here, so I work in prairies, mostly tallgrass prairie, doing these native mycorrhizal inoculants. But people all over the world in different types of ecosystems have tried similar things. You know, in heathlands, shrublands, forests, even into sustainable agriculture. and found similar patterns that our native microbes seem to be the key to improving plant response and the ecosystem function of that soil. So I'm really excited to be a part of this field and do my little part in Prairies. And I wanted to shift gears a little bit and talk about some of my other inoculant endeavors. First, I'm going to talk about my small business Microbloom, and then end with talking about this nonprofit InVam, which I'll explain in a bit. So with regards to Microbloom, this is a company I started about 10 years ago, and it stemmed from me and other people in the lab presenting our awesome work on AM fungi inoculations in restoration and in agriculture, and people being super excited about it, and then asking it like, well, how do we do this? Where do we get it? It's actually really hard to grow fungi, to find fungi in some places that are healthy. So the answers weren't really good, and especially as I started to look at commercial products that were available, and they tend to be quite terrible, globally. The answer was super unsatisfying, like don't buy those, or you can't find native AMF. So after saying that enough times, I decided to start Microbloom, which is my small business which provides native inoculants. And just a little bit more about Microbloom. Was originally started with some NSF, National Science Foundation, and USDA entrepreneurial grants, which taught me a little bit about business and gave me some seed funding to sort of test these bigger scale ideas about inoculants in the world. And what Microbloom sells is a diverse mixture of AMF. They're mostly from prairies. I've got a couple from forest ecosystems in my mix. And these are all local to the US, and I do have ecosystem specific inoculants too. And none of these fungi are in any other commercial products, and I have created custom mixtures for people depending on the situation. Most of the people that use Microbloom are land managers, farmers, growers of all types. It's extremely popular in the cannabis industry. I don't know why, but that is how it is. Yeah, I guess they have more money, maybe. As a scientist, it's been really fun to also have Microbloom because people do science with it, and it's facilitated the science moving forward. So, for example, in the past year, I've worked on some new project as a consultant and providing inoculum for a project in North Dakota where I did a custom North Dakota mycorrhizal blend with them. I got to work in these huge commercial greenhouses where they're growing hundreds of thousands, millions of little seedlings every year. It's been really fun. And I've got to test how my inocular works against battling new weeds like the spartina that they have in Ontario is a big problem. So situations we might not have here. And then there's lots of people who, they buy my product and they do whatever they want with it. And sometimes it's crazy stuff. And so I have application suggestions, but people are going to do what they're going to do, which means using new application techniques and tools and timings, all this different stuff. And of course, every site is a little bit different. And with any application, if they're doing a trial, that's a little bit more information. Both successes and failures, which are both great science. And it's led to new trial ideas. So for example, all those experiments with Johnson County, where we were surface broadcasting the inocula, I did that because I thought it wouldn't work, and people really wanted it to work and use Microbloom a lot like that. And I thought, I don't think this is going to work, but I need to give you some data for that. So now I have some data. Microbloom, we have a website, you can check it out. There's different application suggestions for different contexts, in the garden, so all the way to the scale of field restoration. I guess if you are going to grow cannabis, we have some recommendations for you. But there's also lots of frequently asked questions, a ton of information out there. There's a little store too where you can buy inocula. So that's all I wanted to say about Microbloom, just that we exist. Steven in the back, you want to wave? He's at the little Microbloom booth. You should check it out. We have some science, and you can buy some mycorrhizae over there. The last thing I wanted to talk about was InVam, which is the largest library of arbuscular mycorrhizal fungi on Earth, and it exists here at the University of Kansas, over at the Kansas Biological Survey. And Jim Beaver and Peggy Schultz are here, the curators. Wait. That's what they look like. They're really nice. And so together we take care of this collection. It was started in the 1990s and moved to KU in about 2020. And we have 1,000 isolates, about 1,000, of AMF from all over the world. We have 62 species, and it's widely used in the scientific community and beyond. Our mission is to acquire, propagate, characterize, and maintain germplasm of these AMF in living cultures for preservation and distribution to any person or institution. Our scope. This is a phylogenetic relationship. It's okay if you don't like this. But it shows the whole phylogeny of all these fungi and the number of species we have in each genus. So this is just a highlight that we have, most of the phylogeny of these fungi covered in our library. We have 15 genera, 64 species, and then this figure here is all the countries we have fungi from. We just added Argentina, so that's exciting. So yeah, we have fungi from all over the world. The big challenge with this collection is that they're obligately dependent on plants. So every year we have to grow all of these 1,000 different isolates of fungi out, which means our greenhouse is just covered in plants, and each little plant just has one isolate of AMF. It's tremendous amount of work. We also store these fungi in the meantime at 4 degrees C, but fungi don't necessarily like to live that long in a little bag. So some of these fungi might only last 9 months. It's kind of species specific. So some of these species we have to set up multiple times a year. It's a big pain, but we love it. INVAM functions as a nonprofit service center based out of KU. Historically, it was NSF funded, but it is no longer. The only way it maintains support these days, besides a little bit of facilities from the university, is through this Shop Now button. You can go to INVAM and buy fungi. They are pretty expensive and probably not super useful for most of your at-scale yard needs, but for research, it's really important. And a lot of commercial businesses use this collection to start up their own products, for example. A lot of what INVAM also does is outreach and training and support for researchers and industry. So for example, here's a picture of the Kansas Teachers Summer Institute where Peggy Schultz brought in some high school teachers. We taught them how to do mycorrhizal response assays, inoculation studies, and Washington plant roots. And then they took that and some inocula back to their classes to teach their high school students how to do this. Another huge resource that INVAM has is its website, which isn't very pretty, but there's a ton of great info there. So you can click on methods and find anything you want to know about spores, how to grow or culture mycorrhizae, for example. We also have species descriptions where you can click on different species and look at how pretty they are and some of their spore traits and start to learn identification of these fungi. And the website is widely used. We get about 150,000 users across the globe. For some reason, not so popular. We'll work on that. And to learn a little bit more about INVAM, we have a website. You can contact us via email. As I mentioned, the curators were in this podcast Up From Dust. We were also recently featured in Sierra Magazine, so you can check those out. And someone made a Wikipedia page for us, which is really fun, so we're all on the internet now. So that's about all I have to say about fungi. I'll take some more questions, and then take a picture. For the remaining, I'll answer some questions for a few more minutes, and then you can find me at the microbloom booth. And then there's a microscope table. I'll turn those on, and you could look at some spores if you want, look at some AMF and roots if you want, and hear me crush a spore. It's a single cell, but you can actually hear it break if you have pretty good ears. So I'll be crushing some spores. And Adam is a research technician in the lab. He might be helping me over there. And then Steven will be at the microbloom booth. So any questions?
Yes, about the adverse effect of the weeds and the native plants growing. Do you believe that the fungi had an adverse effect on the weeds, or just that there were so many more native plants that they didn't have space?
Yeah, great question. So when we do the mycorrhizal response assay, so like a plant along with the inocula, sometimes we see some plants having a negative response, but it's not usually that common. Usually they just have none. So what I think is happening with those weeds is it is more the native plants are supportive and they can outcompete the weeds. Yeah, so it's like a trade-off. You get more native late citrus plants and it's a big bushy baptisia and it's shading out your weeds.
But you said somebody was doing research to try and use it against something, right?
Yeah. Mm-hmm. Yeah, so we do that in the lab a lot. We have a lot of weeds that we assess their mycorrhizal relationship. And most of them don't like for AMF. There is one exception, which is really annoying, which is the cuniata sometimes thinks it's okay. Yeah. I know, it's not like
Have you been able to, or is this part of your team to assess soil fertility and the initial mycorrhizal community before you start this? Because I'm wondering whether that might count for some of the unpredictability in responses.
Yeah, good question. It's pretty, so when I'm assessing like success of my treatments, I'm usually looking at plants because it's easy to look at plants. It's actually really hard to look at fungal communities and be like good and bad, even established or not. So things like root colonization, even fungi that aren't beneficial, like our disturbed fungi, can colonize roots and we'll see them there, but that doesn't necessarily mean they're good. So we usually look to the plants as a really easy metric to assess health.
You don't have an easy way to say, okay, this is the initial condition. And then,
And then usually comparing the treatments after. And some of the things we've looked at is like soil aggregate stability across inoculated and not, and like total carbon and inoculated not. And we can see responses at that sort of level. Yeah. But usually I don't assess the original mycorrhizal community. I just assume it's pretty junky because it usually is because we've done a number to our soil. Yeah.
And so, when you converted the brome field, did you do anything besides just plowing it?
Nope, just tilled.
And the other is, I saw one slide that said you were hitting up the prairies that were designated for being taken out. But how else do you harvest good prairies?
Yeah, it's really hard. So, like to start some of these collections, we had to get special permissions from either state entities or, in some cases, private landowners to be able to sample the soil.
So, and then what would you do?
Just like dig a soil core, so you pound it into the earth. You can get, a 10-inch by 2-inch sort of core, and from there we can get enough fungi out of there to start a whole fungal library, and we can grow it up in the greenhouse to bulk propagate it before we put it into restoration. So that's our strategy. We think it works really well, and it reduces the need to like continually harvest from intact native habitat, which I think is not great. We do have a lot of Kansas prairies, but I don't think we should be digging them up for restoration as much as possible. So that's my thought on that.
Have you tried modifying a seed drill so that it's got a second box on each row that would handle the knock?
Like a side pouch? Yeah, I'm not a farm person at all, so I have not done that at all, but that's a fun idea. I know that some of the seed drills do come with like different size compartments, where like even some of the farm tools will have like a fertilization compartment and a seed compartment, so there's some of those things out there, but I've only tested a number of different pieces of equipment so far. But yeah, I think finding a way to put the inoculum and the seed in the same furrow would be the most effective way where you can use at least an amount. Yeah.
Sort of on that note, have you been able to sort of crowdsource any kind of usable ideas or data? You mentioned that you have people, not necessarily applying it according to the directions or using it in non-static measures. Have you come across something that someone has done that has proposed a question for you? Or
Yeah, so I guess there's this project with these folks in North Dakota. I don't remember the state department up there, but instead of doing my application, they did like half, but they didn't necessarily do treatments, and their metric was like, we put it on everything and it's a positive. So like there's a lot of that out there. Like people don't necessarily do like trials in the way I do it with like 5 replicated blocks that are spatially distinct. There's a lot of like, even if they do have a plus minus, it's usually just a 1, and it is kind of hard to make inferences from that. So I just kind of like collectively take in all this data and try to use it in the next trial. Do we want to go for more questions or take a break if anybody wants to leave, and I can answer more questions or meet you at the scope? I know you've been sitting for a long time at this point, so
Let's thank Liz again.
people to check out these